Hello students, in this video you will be studying about the animal kingdom classification. Over a million species of animals have been described, identified, classified till now. These species differ in structure and form. In spite of these differences, there are fundamental features which are common to various individuals, like the arrangement of cells, body symmetry, nature of coelom, patterns of various organs and organs. The study of general animal features reveals evolutionary trends in almost each structure and function. A gradual improvement is noticeable from the lower towards the higher animals. These features are used as the basis of animal classifications. Animals are multicellular heterotrophic eukaryotes, and the cells in their body are of several types which are organized into functional units of increasing complexity. The cells of type are aggregated to serve specific functions, thereby they animals are multicellular heterotrophic eukaryotes. The cells in their body are of several types, and the cells are organized into functional units of progressively increasing complexity. The body of the animals may be made of a group of cells, and a group of cells performing similar function becoming tissues, and a group of tissues together functioning as an organ, then organ system and the organism itself. There are several organ systems, each one is concerned with a separate life function, and all the organ systems together forming the organism. So that is how the levels of organization in the animals. Lower organisms will have simpler cells or tissues, and as you go higher in the level, so you will find complex structures and the functions with different organ systems forming an organism. Animal body shows different levels of structural organization. The first one is the cellular level. It is found in the lower organisms. The body is made of many cells, but the cells do not form tissues. The cells show some division of labor, but cooperate little with one another, and they are independent and can change their form as well as function. So the cellular level of organization. Then this cellular level of organization is found in the lowermost, the Porifera group of animals. Then the second kind is the tissue level. This is found in the coelentrates. Here the body is multicellular, and the cells occur in two distinct layers or two different tissues with specialized cells. Then in most of the animals, the body is multicellular with tissues, and then the tissues will be grouped together forming the organs and the organ systems. So this is how the levels of organization in the animals. Every organism which is multicellular begins its life as a single cell, which is called as the zygote, and the zygote which is formed by the fusion of male and female gamete, which the zygote divides many times and forming a mass of cells, and this group or mass of cells they get arranged into 2 or 3 layers, which are called as the germ layers. So the animals in which the cells are arranged in 2 embryonic layers, the outer ectoderm and the inner endoderm, they are called as the diploblastic animals. Examples are the coelentrates or the cnidarians, where the body, the cells are made into or formed into the outer ectoderm and the inner ectoderm. There may be some mesoglia in between the ectoderm and the endoderm, which is undifferentiated layer which is present between the ectoderm and the endoderm. Then in still higher animals, the developing embryo has got a third germinal layer which is called as the mesoderm. So outer endoderm, middle mesoderm and the inner endoderm. So in between the ectoderm and endoderm there is the mesoderm present, and such animals are called as triploblastic animals. Examples are platyhelminthes onwards. All the higher animals belong to this triploblastic organization. Then comes the symmetry. Every multicellular organism begins its life as a single cell, which is called as the zygote, which is formed after the fertilization. The fusion of male and female gamete, the zygote is formed, which divides many times to form mass of cells. These mass of cells, they get arranged into 2 or 3 layers, which are called as the germinal layers. So the germinal layers are formed during the embryonic development. In some animals, the cells are arranged in 2 embryonic layers, the external ectoderm and the inner endoderm. So such animals are called as diploblastic animals. The coelentrates are examples of diploblastic animals. And there is an undifferentiated layer which is called as the mesoglia present between the ectoderm and the endoderm. There is another group of animals in which the developing embryo has got a third layer, third germinal layer, which is called as the mesoderm, which is seen in between the ectoderm and the endoderm. Examples are platyhelminthes onwards. All the higher animals, they do have this kind of organization. Animals can be categorized on the basis of their symmetry. Symmetry means the similarity in the arrangement of parts on the opposite sides of the body. So the similarity in the shape, size and the number of parts on opposite sides of a median line is symmetry. And the median line is the plane through which the animal can be cut into 2 equal halves. Now in some animals, the body cannot be cut into 2 equal halves through any plane which is passing through. So such an animal is called as asymmetrical. So sponges are an example. So any plane that passes through the center does not divide the animal into 2 equal halves. So you cannot make 2 equal halves in case of an asymmetrical animal. Sponges and snails are examples. Then there are other animals where the body can be divided into 2 identical halves through any plane which is passing through the central axis of the body. So such a symmetry is called radial symmetry. So the animals like hydra, jellyfish, they have a body with a number of similar parts which are radiating out from the central axis. So the body can be divided into equal halves by any plane which is passing through the center from top to bottom. So such a symmetry is called the radial symmetry. Then there is another kind of symmetry in case of frog, fish and higher animals. The animal can be divided into two equal halves by only one median plane. So you will get the left side and the right side, or left half and the right half. So the division is possible only in one plane. Such a symmetry is called as bilateral symmetry. Any two halves you get, either the left and the right. So two halves are possible. So such symmetry is called bilateral symmetry. You will find this kind of symmetry in fish, frog and goat. Here you can see the diagrammatic representation of asymmetry, where no special shape or pattern is there in the body. And the second one is radial symmetry. The body can be divided into identical halves by any plane which is passing through the center from top to bottom. So that is radial symmetry. And the last one, where the body can be divided into the left half or the right half, which are identical. So that is bilateral symmetry. The presence or absence of a cavity between the body wall and the gut wall is very important in classification. The body cavity, which is actually the space between the body wall and the alimentary canal, which is actually produced by the splitting of the mesoderm during the embryonic development. Now, animals which do not have any body cavity are called as acoelomates, and in them the space between the body wall and the alimentary canal is filled with parenchymatous cells or tissues. So example is platyhelminth. So there you will not find the space between the body wall and the alimentary canal. Actually that area is filled with parenchyma. Then the second type is pseudocoelomate. These animals show a false body cavity. In roundworms, the body cavity is not lined by the mesoderm, which is termed as the false coelom or the pseudocoelom. Instead of the lining, you have the body cavity, which is lined by patches of mesodermal cells. At certain regions only you will find the mesodermal cells. So they are called as the pseudocoel or the false coelom. So those animals which are having a pseudocoel are called as the pseudocoelomates. Aschelminthes are the examples of pseudocoelomates. Then there is the third kind, where you will find a true body cavity. The true body cavity is called as the coelom, and this is present in the triploblastic animals. So here during the embryonic development, the mesoderm splits into two layers enclosing the body cavity. So these two layers will be lining the inner side of the ectoderm and the outer side of the endoderm. So thereby the coelom is formed. Cavity is filled with a fluid which is called as the coelomic fluid. In some animals like cockroach, the coelom is filled with blood. So when blood is there in the coelomic space, it is called as hemocoel. Arthropods and mollusks, you will find the true coelom, which is you will find the true coelom greatly reduced, but it is filled with blood, or blood spaces are there, which forms the hemocoel. In this diagram, you can see the acoelomate, pseudocoelomate and coelomate body cavities. In the acoelomate structure, you can see the ectoderm, which is given in blue color, and the endoderm, which is in the yellow color. So everywhere it is given ectoderm blue and endoderm yellow color, and in between you will find the pink colored cells. Those are the mesodermal cells. Okay, so in acoelomate animals, the space between the ectoderm and the endoderm is filled with the mesoderm. But in pseudocoelomate, you will not find the complete layer of the mesoderm, but mesoderm will be seen at certain places as patches, which is called as the pseudocoelomate. And in the coelomates, where you will find the ectoderm and the endoderm, but in between the ectoderm and the endoderm, that is inner side of the ectoderm and the outer side of the ectoderm, are lined by the mesodermal cells. Thereby it forms the true coelom. In some animals, the body is externally and internally divided into segments with a series of repetitions of at least some organs. For example, the earthworm, the body shows this pattern, which is called as metameric segmentation. So this phenomenon is called metamerism. So repetition of parts in an animal's body, and this segmentation occurs in highly organized phyla like Annelida, Arthropoda and Chordata. Each segment of the body is called as a metamere, and segmentation often affects both internal as well as external structures. So that is metameric segmentation. And in chordates, the segmentation is seen only in the embryonic stage. Then notochord is a mesodermally derived rod-like structure which is formed on the dorsal side during the embryonic development in some animals. So animals with the notochord are called as the chordates, and those animals without the notochord, or they do not have the notochord, they are called as non-chordates. So phylum Porifera to phylum Echinoderms are the non-chordates, and the rest of the animals are having notochord, so they belong to the group Chordata. The animal kingdom is divided into 11 phyla according to the levels of organization, their symmetry, the body cavity or the coelom. On the basis of these characteristics, the animal kingdom is classified into 11 phyla. The various phyla are: the first one, phylum Porifera, then phylum Coelenterata or Cnidaria, then third one is Ctenophora, then comes Platyhelminthes, Esculminthus, Annelida, Arthropoda, Mollusca, Echinodermata, Hemichordata and Chordata. All these are the various phyla in the animal kingdom. Now you will be studying about each phylum in detail with the characteristics and their examples. Here you can see the examples Cycon and Euspongia. No specific shape also for them. And in the next diagram you can see the body of the sponge, the body wall, and the body wall is having the what you call spongiocele inside the body cavity, which is the spongiocele, and the spongiocele is lined by the flagellated cells, which are the choanocytes, and there are openings which are called as the ostia, smaller openings on the body wall, which are the ostia, bringing in the water to the spongocoel, and the water will escape out through the terminal opening, which is the osculum. Now when the water passes through the ostia to the canals into the spongocoel, and as the water moves out of the spongocoel, there would be exchange of gases and collection of food, and all those processes will take place. And mouth and digestive cavities are lacking, but they do have intracellular digestion. That means particles can be taken into the cell and there it is digested in the food vacuoles, like the case of amoeba. Now these choanocytes are specialized cells which are lining the spongocoel. They are with flagellum also. By the beating of the flagellum will cause the movement in the water, and thereby the water can be going out or escaping out through the osculum. Now the sponges possess internal skeleton made of tiny calcareous or siliceous spicules. The internal skeleton is called as spicules, or they are called as spongin fibers. Now these spicules and spongin fibers are present or located in the mesoglia or the mesenchyme, and the spicules and the spongin fibers are secreted by the cells in the mesenchyme, and these internal skeleton are formed of protein collagen, and they occur as a network. Now the reproduction in sponges or porifera, they have asexual reproduction as well as sexual reproduction. Asexual reproduction is by fragmentation as well as by budding. Now by budding, special cells are formed which are called as the gemmules, and the gemmules will get separated from the parent body, and another new sponge will be developed. We will study about the first phylum, that is phylum Porifera, the sponges. The Porifera includes very strange lifeless and plant-like creatures which are commonly called as the sponges, and the body of the sponges are vase-like, cylindrical, but majority form flat or irregular branching masses. So the symmetry of the sponges is asymmetry. They do not have a specific shape, so their body is asymmetrical, and the sponges develop from two germ layers only, so they are diploblastic animals. And most of these sponges or phylum Porifera are marine animals. Some of them are freshwater. The body shows a cell aggregation plan, no tissues or organs. The body wall encloses a large cavity which is called as the spongocoel. Spongocoel is the body cavity, and the spongocoel is having openings which are smaller in size, which are many in number also, which are called as the ostia, and there is a bigger opening which is leaving the spongocoel, which is called as the osculum. So the sponge body wall is enclosing the cavity which is called as the spongocoel, and the spongocoel in the higher sponges, they do contain numerous small cavities on the body wall itself, which are called as the canals, Now either the spongocoel or these canals are lined by specialized cells which are called as the choanocytes. So these choanocytes are the cells with flagella, and because of the beatings of the flagella, it maintains a steady current of water through the canals in the sponge body, and the water current enters through the small pores which are called as the ostia, and um, the water current comes in through the ostia, and it passes through various canals and enters into the spongocoel, and finally leaves through the larger aperture which is called as the osculum, or the plural is osculum. Sexes are not separate. That means these sponges are hermaphrodites. Both male and female sex organs are present in the same individual. So the sponges, they produce eggs and sperms, which are produced by the same individual. And the sperms leave one sponge and enter into another sponge with or through the water current, and fertilize the eggs inside the spongocoel or inside the sponge body. So the fertilization is called internal fertilization. The zygote develops into a new sponge by holoblastic segmentation, and the life cycle includes a free swimming larva, which is the amphiblastula or the parenchyma. Two types of larval forms are there in Porifera, amphiblastula and parenchyma, and the larva escapes through the osculum with the outgoing current of water. And there are about 9,000 species of sponges live at present. The second phylum, that is phylum Cnidaria or phylum Coelenterata, includes all jellyfishes, sea anemones, corals. The phylum Cnidaria or Coelenterata are aquatic, mostly marine. They are free swimming or attached ones, sessile, and they are radially symmetrical animals. Okay, there are colonial forms also. They show two main types of individuals or the zooids, the polyp and medusa. The polyp is cylindrical and which is normally a sessile one, fixed one, but maybe solitary single ones or live in groups or colonies. The medusa is basically umbrella shaped or umbrella like, and of course free swimming, and either or both the forms may occur in a species. The polyp and medusa may be seen in the same species, then such a condition is called the polymorphism, where both polyp and medusa forms are found in the same animal. The two forms alternate with each other in the life cycle. This is like the sporophytic and the gametophytic generation of the plants which you have studied earlier. Here the polyp and medusa, they alternate in the life cycle. So alternation of generation or metagenesis is there in the phylum Cnidaria, which are the sac animals. Symmetry is usually radial. So the upper oral side and the lower, which is the attached side. Then the Cnidarians, they develop from two germ layers only, ectoderm and endoderm. So they are diploblastic organisms like the previous phylum, that is phylum sponges. And these Cnidarians, they have tissues but they lack organs and organ systems. So they have tissue level of organization. The body wall consisting of two distinct layers of cells, the outer epidermis and the inner gastrodermis. And each layer has several types of cells which cooperate with one another as in tissues. So the cells include the stinging cells or the cnidoblasts, and they are for offense and defense. You can see the nematocyst or the nematoblast, which are used for defense. Now between the two layers, that is between the ectoderm and the endoderm, there is a sheet of gelatinous material which is called as the mesoglia. Now the stinging cells or the nematocyst, when discharged, give out from the sac, the cnidocyst or the nematocyst, a long thread like tube which may coil around the prey, and it produces a toxin. By injecting the toxin to the prey, it can even kill the aquatic organisms. The body encloses a single body cavity which is called as the coelentron, and similar to the spongiocele of the sponges. Here the body is enclosed in the cavity which is called as the coelentron. And the coelentron is having only one aperture which is called as the mouth. There is no anus. The mouth will serve both the purposes, taking in food as well as throwing out the excretory or the waste materials. So an incomplete digestive system, only one opening, that is the mouth. And the digestion of the food is partly intercellular, which occurs in the coelentron in the body cavity, and partly intracellular, like the food vacuoles in the sponges or in the amoeba. Reproduction for this cnidarians, the sexes may be separate, or you may find both the sexes in the same organism also. Reproduction is usually by asexual means, that is budding of the polyp, and sexual reproduction in the medusa form. Alright, and the life history includes a free swimming larva which is called as the planula larva, which is ciliated. And there are about 10,000 species of living cnidarians or coelentrates. So the cnidarians, they have a tissue level of organization, special stinging cells called as the cnidocytes or cnidoblasts are there for defense and offense. Then incomplete digestive tract bounded by the body wall, that is the body cavity coelentron with only one opening, which is the mouth. Then simple gonads are there in this coelentrates. Here you can see the coelentrates, sea anemone and the jellyfish, both are shown down, and the upper figures are of the polyp and the medusa. Okay, and the polyps are usually sedentary ones, attached ones, and the medusa are the free floating ones, and they both of them have the tentacles. With the help of the tentacles, the movement is possible for the free living medusa, and the medusa has got the reproductive structures. So you can see the mouth and the gastrovascular cavity or the coelentron. Now the down one is the sea anemone, where you can see the mouth is there, but which is surrounded by a group of tentacles, and this sea anemone is an attached one. In this photograph, you can see variously colored cnidarians at the bottom of the base of the sea. So differently shaped, differently colored, different species of coelentrates are seen at the bottom of the sea, and their body, as the polyps and the medusae die, they become solidified because of the deposition of calcium carbonate, and as they live in groups or colonies, and when those old ones die, the young ones will be formed, which will be with the colors, and the dying old ones will become calcareous, what do we call, the coral reefs, that is smaller hilly continuous long continuous hilly regions made of the depositions of the coelentrates forming the coral reef. So the corals are formed from this kind of coelentrates at the base of the sea, or at the bottom of the sea.